In this video, we're going to be learning how the stomach performs its primary function, that being the chemical digestion of food, more specifically the chemical digestion of protein. Now, as expected, this is not the role of the physical structure of the stomach, but is actually performed by the chemicals found in the liquid part of the stomach, which is what we call your gastric juice. And gastric juice contains three specific components, all of which play an important role in the chemical digestion of protein, starting with water that makes up most of the liquid volume of gastric juice. Now, water in gastric juice fulfills the same role as water in saliva does, acting as a solvent to dissolve the partially digested food that ends up in the stomach, making it easier to digest. Now, the other important role that water plays is in its assistance of the actual digestive enzyme found within the stomach, which is a digestive enzyme that we call pepsinogen. Now, remember from our introduction that digestive enzymes catalyze what we call hydrolysis reactions, where the enzyme adds water to the chemical structure of a large nutrient in order to break it down into smaller nutrients, which in this case are amino acids. Therefore, this digestive enzyme would not be able to perform this function if water was not present in the stomach. Now, assisting the water in the dissolving of any food that makes it into the stomach is highly strong and concentrated acid, hydrochloric acid, and this generally makes the entire dissolving process much easier because of the effect that acid has on proteins. So if we take a look at the 3D structure of this protein here, we can see that its folding is very complicated, which makes it difficult for pepsinogen to actually get into the center of this protein and digest it. However, we know that strong acids such as HCl can disrupt the 3D structure of this enzyme in order to cause the protein to lose its folding like this. Once the 3D structure of the protein is unfolded, this makes it much more easier for a digestive enzyme like pepsinogen to cut the protein apart into small individual amino acids. This process, of course, is what we call protein denaturation, so we can say that the acid has effectively denatured any of the protein components of the food that make it into the stomach, greatly speeding up the process of digestion. Now, the other thing that HCl does is that it actually functions as a way for pepsinogen to become active or inactive by acting as effectively a chemical on-off switch for the enzyme that is responsible for digestion in the stomach. Now, the obvious question here is, other than how does HCl do this, is why is this chemical on-off switch actually necessary? Well, let's talk about what pepsinogen does. So pepsinogen, as previously mentioned, is the primary digestive enzyme of the stomach and functions as a protease, meaning that its primary role is to digest the protein component of food. Now, the problem that we need to consider about pepsinogen is what pepsinogen does if the stomach is empty. Well, if the stomach is empty, there is no food in the stomach, meaning that there is no protein in the stomach for this enzyme to break down. Now, the problem in the case of pepsinogen is if we consider what is found inside of the walls of the stomach, we remember from the previous video that the stomach has the ability to contract in order to compress the food within it. This is an important part of the stomach's mechanical digestion, meaning that there is muscle found within the walls of the stomach, and muscle, of course, is made entirely of protein. So if the stomach is empty, this means that pepsinogen, an enzyme that breaks down protein, is actually able to attack the muscle in the walls of the stomach and digest the protein components of the stomach walls. This is obviously very bad, and there are a host of medical problems that occur when the stomach walls actually begin to get digested and burned from the acid as well. And as a consequence of this, pepsinogen is one of the most tightly regulated enzymes within the human body. For starters, pepsinogen is not actually the active form of the enzyme. Pepsinogen is actually the form of the digestive enzyme when it is in the off position, if you will. However, when food, specifically food that is rich in protein, enters the stomach, your nervous system sends a signal to the cells in the wall of the stomach to start releasing HCl. And this HCl is going to interact with the structure of pepsinogen and cause the folding on the structure to change, thereby opening up the primary location where pepsinogen functions, which is, of course, the enzyme's active site. Now, when this structure happens, this is what we call an alteration in conformation, or simply a conformational change, where the folding in the structure changes in response to the HCl in the stomach, opening up part of pepsinogen's structure. And when the active site opens up, the active site is actually able, because pepsinogen is a protease, is actually able to digest part of the structure of pepsinogen itself, cutting this part of the protein off, which is what we call its masking sequence, allowing the active site to now be fully open without this piece of the protein in place. And now this form of pepsinogen, which is now what we call pepsin, is now fully active and able to begin digesting protein that we find within the stomach. Now, eventually, pepsin, the active form of the enzyme, is going to run out of protein to digest, so even before we manage to switch the enzyme off by decreasing the amount of HCl in the stomach also needs a very, very protective structure in order to prevent the muscular layer in the stomach, which we actually find in here, from being targeted. And we can see when we look at the structure of the stomach walls that the stomach walls have many protective layers that protect the muscle on the inside from being targeted by active pepsin and also by the hydrochloric acid on the inside. So the first of these protective layers that physically prevents pepsin from being able to get at the muscles is actually a layer made entirely of mucus. So mucus occurs in many different locations within the body, most notably up the nasal cavity inside of your nostrils. If you take your favorite finger and stick it up your favorite nostrils, you can feel what mucus feels like. And most people would describe it as feeling wet, because water is obviously a major component of mucus. So within the mucus layer, there is going to be a large amount of water, and this is going to be useful in terms of reducing the effects of the HCl found within the stomach. Most people have the understanding if you want to take a dangerous acid and make it less dangerous, you can add water to it in order to decrease the concentration of the acid and therefore make it weaker. Effectively, the water within the mucus layer is going to dilute the acid found within the stomach and make it significantly less corrosive. Now, the other obvious physical barrier that the mucus layer provides is because the mucus layer is made entirely of carbohydrates, aside from the water that is found within. Now, the big barrier here is that carbohydrates are not proteins, which means that the pepsin, which is a digestive enzyme that is specifically built to target proteins, is not able to digest the carbohydrate layer that the mucus is made of, meaning that as long as this mucus layer is here, pepsin is not able to pass through it and not able to digest the muscle that is found within the inner layers of the stomach walls, and through a combination of the carbohydrate layer and water found within the mucus layer, it protects against both acidic digestion and enzymatic digestion that is able to reach the inside. In part 2 of this video, we're going to look at more complicated measures of how the inside of the stomach protects itself from digestion, as well as look at the negative consequences of when these protective measures are not actually enough, and what consequences can happen if the stomach wall is actually digested by its own acid and digestive enzymes.